Hello everyone, my name is Salinda Yadav of class 2 RBC Division C of roll number 649 RJ College of year 2022-2023. So let's start. Today my topic is total RBC count. RBC count are red blood corpuscles which are also called erythrocytes. So red blood cells or erythrocytes are the most abundant cells in the blood stream and contains hemoglobin, the compound that carries oxygen through the body which require for human being. Any destruction of red blood cell, its quantity, shape, size, structure or life cycle can therefore affect the oxygen carrying capacity of the blood and affect the human organ body. RBC shape and size. Normally disc shaped and biconcave shape, central portion thinner and periphery thicker. Size diameter 7.2 micron, thickness at periphery 2.2 micron and end center 1 micron, as you can see in the image. So what a method of RBC count? There are two methods: visual hemocytometer, which are also called manual method, and automated method. So manual method have types: Neubauer chamber, RBC pipette, RBC dilution fluid, EDTA sample. These are components used in the manual method. What are RBC dilution fluid? RBCs are around only 5 million cubic millimeter of blood. Counting this much number is highly impossible. Therefore, the blood sample is diluted with the help of RBC dilution fluid. It fixes and preserves RBC. It is isotonic to RBC, which are required for the solution. So the most frequently used hemocytometer in the RBC count use is the Neubauer chamber. Their size of glass is like as you can see in the image 30 by 74 millimeter. The rule area is 3 millimeter to divided into 9 large squares, each with 1 millimeter square area. The large central square, which we seen in the entirely with the 10x objective, is divided into 40 medium square with double or triple lines. As you can see, there are grid lines of total RBC count we use. Each is 25 squares are again divided into 16 small squares with single lines, so that each of the smallest square has an area 1 by 400 millimeter square. As you can see in the image, there are grid lines how RBC counting area are absorbed. The last center square is used for RBC count. Has already stated, these areas are divided into 25 medium squares, which in turn are each divided into 16 squares. 25 medium squares, only the 4 corner squares and the center squares within large center square are used to perform RBC count. With a pipette, carefully draw off around 20 millimeter of the cell mixture dilution. Place the pipette tip against the edge of covered glass and slowly expel the liquid until the counting chamber is full. Capillary action with the help to ensure that the counting chamber is full, but care should be taken not to overfill the chamber. A volume of 10 millimeter is sufficient to fill one counting chamber. So there are how the RBC count are done. Count the RBC. The microscope must be switched to photo subjective for better counting. Count the cells in the respective areas as stated earlier. Write down the amount of cell counted with the help of the microscope. So there are dilution fluid types: Hames fluid and gaseous fluid. The composition of Hames fluid are sodium chloride 0.5 gram, sodium sulfate 2.5 gram, mercury chloride 0.25 gram, distilled water 100 ml. And the composition of gaseous fluid: disodium citrate 3 gram, formalin 1 ml, distilled water 90 ml. This dilution fluid is cheap and commonly used in the total RBC count for better observation. What a procedure to count the total RBC count. So let's start. Assemble all the equipment. Draw the blood directly from finger or collected sample into RBC pipette up to 0.5 mark. Wipe up the tip of the pipette to remove extra blood if present. Then immediately draw the dilution fluid up to 101 mark. Now rotate the pipette gently for 2 to 3 minutes so that the dilution fluid gets mixed properly. This will give dilution 1:200. Place the cover slip in position over the rule area of chamber. Once again mix the solution thoroughly by rotating the pipette. Discard first 1-2 drop of blood from the pipette. Now apply slight pressure on the rubber tube of pipette so that the third drop of fluid is hanging position. Touch the tip of the pipette against the edge of a cover slip. The angle between pipette and the cover is 45 angle. With this process, chamber gets filled with the fluid. This is known as charging of the chamber. The care should be taken that no air bubble is present inside the chamber and there is no cover filling beyond the rule area. Now count the RBC using 40x in central square. The central square is subdivided into 25 squares. Out of 25 square, count 4 at each corner and 1 at center. Each of these 5 square is subdivided into 16 small squares. Thus RBCs are counted in 16 multiplied by 5 equal to 18 small squares. In case of margin cells, count the cells on L line, either right and lower or left and upper margin. Make the total of cells counted in 5 squares. As you can see in the image, the grid lines of RBC count. The errors of RBC count. There are technical errors, inherent errors. Technical errors can be minimized by use of calibrated apparatus and by careful technique. The distribution of and the inherent error. The distribution of RBC of an irregular pattern in the counting chamber. This affects the accuracy of usual count, which can be minimized by counting a large number of RBCs. There are differences in adult. RBCs are 4.5 to 5.5 million cubic millimeter, and in adult female 3.8 to 5.2 million cubic millimeter, and at the birth 6 to 8 million cubic millimeter. Thank you.